Hello, and welcome to my presentation on what is visual phasing, the general case versus the specific cases. And this is going to be part of a 2 or 3 video series. They may not all be consecutive. I may make some videos on some other topics in between. The goal of these videos is to clear up a little bit of the terminology that people use in, say, Facebook groups when they're talking about things like visual phasing and phasing, so that we're all on the same page, and that when someone says something like you can phase a parent using a child, and then the other person doesn't say that's impossible because they're only thinking of the specific case of parent phasing. Or when someone says you could visual phase without siblings, and then people look at you funny because they're only thinking of the specific case. For example, maybe they're thinking of the Stephen Fox spreadsheet type of visual phasing. But these terms are actually a little bit more broad than we're using. And I want to start out with the general case with visual phasing. So my definition of visual phasing, and this is the general case, is using a visual representation of a DNA segment data to assign parental phase to autosomal and or X chromosome blocks. That's a lot of big words there. Using a visual representation of DNA segment data to assign parental phase to autosomal and or X chromosome blocks. So let's break that down a little bit. Basic visual phasing terminology. There are 4 terms really used in the definition, or 4 main concepts. Autosomal chromosomes, what are they? DNA blocks and segments, visual representation, and parental phase. What does it mean to assign parental phase? And some of the stuff, I mean, there's going to be viewers here of all levels, so some of the stuff is basic, so bear with me. And on the other hand, some of the stuff might go over people's heads, so bear with me there as well. Hopefully there's something for everyone in this presentation to get us all closer to the same page. So successful visual phasing requires familiarity with all 4 of these components of the definition. So we're going to look at each one in detail in this presentation and go over the difference between the general case versus at least the most popular specific case, the Stephen Fox visual phasing spreadsheet, which is an excellent tool, as well as the difference between visual phasing and actual phasing, because I think a lot of people don't know that there's a difference or don't know what the difference is starting out. I call this slide 46 and you, because you always hear from like 23andMe that we have 23 chromosomes, or when you go to Jedmatch, it shows you chromosomes 1 to 23, but you're actually looking at 46 chromosomes. It's just the way we number them, that it's we number them by pairs. And this is a karyotype of human, the chromosomes in a human nucleus. And you'll see they start out, we number them by their length. So chromosome 1 is much larger than, say, chromosome 22. 1 through 22 are called the autosomes, and they're your regular chromosomes. 23 is your Y and your X, and we call them the allosomes. And if you're a male, you'll have a Y and an X, and if you're a female, you'll have an X and an X. I mean, there are some exceptions and some other issues with that, but the general rule is a male gets a Y from his father, and that's why he's a male. And whereas a female will get an X from both the father and the mother. That's the fundamental difference between the allosomes and the autosomes. So regarding each of these 23 chromosome pairs, we inherit 1 copy of the chromosome from our father and 1 from our mother. And the chromosomes are numbered by the pair to which they belong and not individually. So there's not chromosomes 1 through 46. There's just chromosome pair 1, and we often refer to that simply as chromosome 1 as when we speak loosely. So by this convention, we each have what is referred to a paternal copy of, say, chromosome 11 and a maternal copy of chromosome 11. And that's how we typically refer to them when we want to distinguish between the two. So chromosome pairs 1 through 22 consist of the chromosomes called autosomes, and chromosome pair 23 consists of the 2 allosomes, the X and the Y chromosomes. And this distinction is critical because they have different inheritance rules for the allosomes. And those inheritance rules will vary by gender, as I explained. Like a father can only pass a Y to a son. Otherwise, if he passed an X, it would have been a daughter. Okay. Next thing we need to know is DNA is digital. And by digital, I mean it's more like ones and zeros and discrete, finite blocks and segments, as opposed to, say, a solution where, if you mix two people's DNA, it just dilutes. DNA is not like that at all. It is far more analogous to digital. So DNA is inherited in blocks and segments. Then these are almost like chapters and scenes on a DVD. Furthermore, each copy of the chromosome can be divided into a countable number of base pairs. It's a molecular unit of a DNA molecule, and each of these base pairs consists of four discrete possible values that correspond to the four types of nucleobases found in a DNA molecule. When you open up your raw DNA file, you'll see A, C, G's, and T's everywhere, and that's what I'm referring to. That's the actual data collected at each position by the testing companies. And the A stands for adenine, the C for cytosine, the G for guanine, and the T for thymine. And these are the nucleobases of a DNA molecule. You really don't need to know for DNA analysis, the names of these bases and things like that. But what you do need to know is that there are four possible choices, that each base pair, you'll have two of these choices. Base come in pairs on a chromosome because of the double-stranded helix structure of DNA molecule. And I'm not talking about the pair, the A, C, reported in your raw data file. One of those is paternal on your paternal copy and one's on your maternal copy. What I'm talking about is within the paternal copy, for example, there are two strands. Only the base in the positive coding strand, that's what it's called. It's one of the strands. It's the strand that codes by interacting with proteins in a biological situation. But only the base in the positive strand is measured by commercial testing companies. So that's what you'll see in both columns of the raw DNA file. You'll see the positive strand value for the paternal and the positive strand value for the maternal. The base on the opposite negative strand for each of these could easily be computed from the value of the base in the positive strand. So there's no need to measure to or report to, and the positive strand is easier to measure because it interacts easily with things that you can measure them with. So a segment of DNA typically consists of several million base pairs, and we're talking about the length of the segment now. So we use the unit megabase pair to measure the digital or positional length of a segment, just as we use megabytes to measure the size of a file on a CD or DVD. And a byte is something you can actually count. It's a 1 or a 0. Well, actually, a bit is a 1 or a 0. It's a certain number of those, but it doesn't matter. It's the same idea. And a base pair is the fundamental unit of a DNA molecule. So that's some background on the science of DNA. But the most important thing to get out of this is that DNA is digital. And let's take a look at some examples of how and why this is important. This is probably the most important slide in the presentation to understand. And what we're looking at here, we'll take a look at the picture first because I think it's a little more intuitive than the language on the right, but we'll hit both. What we're looking at is two generations of meiosis, which is cell division, and it's how DNA. It's the process by which DNA is passed down from parent to child, and then in this case from grandparent to father to child. So what we got in the first row, where we start out, we have four, we'll call them copies of chromosome 11, and that represents the copies in two specific people in this example, and that is in the child, who's at the bottom, two paternal grandparents. So the yellow and red chromosomes are the paternal and maternal copies of the paternal grandfather's chromosome 11, and the blue and the green are the paternal and maternal copies within the paternal grandmother of the child at the bottom. And I color-coded each one with a solid color to explain how DNA is digital. The next generation represents the father of the child at the bottom. And let's take a look at the the paternal copy of the father, which is the yellow and red uh, segmented chromosome on the second row in the left side. So the father received DNA from each of his parents, and the yellow-red copy is only referring to the copy the father received from his father, from the paternal grandfather. So the paternal grandfather had a yellow chromosome entirely and a red chromosome copy entirely also, but he only passed down half of his DNA to his son. So DNA is digital in that you had some yellow and you had some red. It doesn't just mix together into a solution and you get some shade of orange. Rather, DNA is inherited in blocks. And what he does on this particular chromosome, the beginning of the chromosome, if you will, the grandfather passes down his paternal copy for a while, the yellow part, and he starts passing down some red. There's a break, and then a new block starts, and he passes down some red from his mother, from his maternal copy to the father. And the same thing's going on the father's maternal side with the blue and the green from the paternal grandmother. So she's passed down some of her maternal DNA for a while in block one, and then she switched or recombined and passed some of her dad's DNA down to the father, and that's the blue, and that's block two, and then she switched again, and then went back to passing down some of her mother's DNA. So still, though, everything's either yellow or red or blue and green. There's no mixtures. There's no, there are absolute points at where this happened along the chromosome. And there are a total of 4 streams of DNA, both at the first level of the generation and at the second generation. And the child's generation, we're going to see a recombination of all 4 colors, because dad is going to pass down some of his red and yellow, and he's going to pass down some of his blue and green. And a blue and green combined section will be a block, and then he'll switch and pass whatever he has in his yellow-red side. So in this example, the father starts on his maternal copy, starts passing the child his maternal copy, which is green at the top and then goes blue. And then he stops. That's one block, although that block has 2 segments. The next block is when he switches and the father starts passing down his paternal copy, which is yellow and red. And within that, because the father had 2 blocks, had a block division there, a recombination, there's going to be a segment division in the child, whereas there's only 2 blocks, 1 and 2, because the father only recombined once in that generation. There are going to be a total of 4 segments in the child, corresponding to the 4 colors in this case, the 4 streams of DNA. And it could be a lot more complicated than this. I mean, there could be a chromosome with 50 segments on it, and they may be from various generations going beyond the grandparent level, of course. But this is a very simple example, but I think it demonstrates the block and segment nature of DNA inheritance. All right, so now let's take a look at the wording on the right, and I tried to formalize it a bit. A parent passes half of his or her DNA to a child through a process called meiosis. So each parent has 2 copies of each chromosome. The parent does not always pass full copies of a chromosome to the child corresponding to the child's grandchildren. That is, they don't pass the whole chromosome down, the whole copy down. Sometimes they do, but not every time. That's the exception rather than the norm. Rather, the stream of DNA being passed tends to break a few times on each chromosome, and the parent begins passing DNA from the opposite grandparent to the child, which then reattaches itself or recombines to the preceding stream from the opposite grandparent. When evaluating the positions of recombination events, we call them recombination points, on a single chromosome in a single generation of meiosis, the span of DNA between 2 consecutive recombination points, or from the tip of the chromosome to the nearest recombination point, if it's a block on the end, we call it a block. We can also detect and measure recombination points in a child from previous generations of meiosis that occurred in grandparents and more distant ancestors. A span of DNA between any two detectable recombination points and or chromosome tips, if they're the ones on the end, irrespective of the generation in which the recombination occurred, or whether there exists intermediate recombination points, is called a segment. And I think the picture explains it a lot better, but I also wanted to put down some formal definitions because I know a lot of the stuff we do in the Facebook groups and things like that is textual responses to people. But I think this is a nice visual here that I think explains the digital nature of DNA inheritance. So let's delve into this model a little further on this slide. Here what we have are two people that descend from those same grandparents. They're first cousins. Um, so cousin one is the one we looked at already and had 4 segments and on his paternal copy of his chromosome. On the other copy on his mother's side, he's not related to this particular other cousin here because this is a paternal cousin. In this example, cousin one's father and cousin two's mother are siblings. That's what I picked here. So then we're going to compare that to the maternal copy of cousin two. So she inherited a different series of segments from her mother, but it's the same 4 streams, although she only inherited 3 of them just by random chance. But they're the same 4 possible colors in this example because they're descending from the same 4 common ancestors, right, or the same 2 common ancestor pairs. So what we have is only a single overlapping color, or where segments of the same color happen to overlap each other, and that's the blue. Cousin one has a little sliver of blue there in the middle, but it happens to be at the same place on the chromosome as cousin two. So we're talking about positional location, and that's how we determine a matching segment. And that's how Ancestry.com, for example, determines whether or not you match your cousin. If these segments that are identical because they came from the same source overlap in position. So even though they have all of the same grandparents on this particular chromosome, they don't match on the whole chromosome because they inherited different parts, different traits, different genes from each of the grandparents, and there's only some overlap. And that overlap is called a matching segment. So now let's formalize that a little bit with the text on the right here. When comparing 2 individuals, a matching segment refers to the overlapping portion of a segment on one individual and a segment on the other individual, where each sampled base measured across the overlap is identical in both individuals. An endpoint of the matching segment can either be the tip of a chromosome or will be at a boundary of a segment on either individual's chromosome. And here the boundaries are segment boundaries on cousin one, right? Because it's a small segment of blue within a larger overlapping segment of blue on cousin two. And this assumes that the matching segment is truly identical by descent. Sometimes, due to miscalls by the testing companies, random mutations, and microdeletions, there may be a few bases that are not exactly identical between these individuals, even though it's a true matching segment. And that's why Jedmatch has things like mismatch bundling thresholds and stuff like that. But in theory, they would be identical because they came from the same stream of DNA originally. But there may have been some mutations and some miscalls by testing companies that may throw in a little a wrong data point every so many hundred calls, and we have algorithms to deal with that. So also it should be noted that the tips of matching segments are typically not IBD. And by tips, this is what we say when we say fuzzy edges of segments. So the matching segment, in theoretical matching segment here, is that one blue part where they overlap. However, when you actually went to go measure it, it may be a little bigger because it may have some fuzzy tips on the end, um, especially if you're dealing with phased data, with unphased data. I'm sorry, because in unphased data, we'll get into that, but using unphased data causes long fuzzy tips on the end of each segment, and a matching segment will usually therefore extend slightly beyond the true recombination point when measured. Okay, so next slide. Let's take a look at some of the common visual representations. Remember, that's the next term in our definition of visual phasing in the general case. Let's take a look at some of the common visual representations of segment data. Some are more visual than others, but visual representations can be in the form of lists, charts, spreadsheets, diagrams, you name it. Any way you can think of visualizing segment data, it's probably been done, and there'll probably be some more new ones coming up too, actually. But you could use any of those to perform visual phasing per the definition. So let's take a look at some of the popular ones that people are using in actual real life visual phasing in the general case. The first tool that everybody should get to know is Jedmatch's free one-to-one compare tool. And these are the types of visualizations of segment data that you get from that. And here you're comparing two people, or two donors, you're comparing their DNA, and it shows you, in this example, there's 7 matching segments, and it gives you the chromosome on which the match exists. It doesn't tell you whether it's the paternal or maternal copy, unless you're dealing with phase data. So you don't know that in this example, unless you know the actual relation between the two donors. So you know the start location and end location, which is measured in base pairs here. But you see it's like 82 million to 101 million. I usually use mega base pairs so I don't have to carry out as many digits, since edges are kind of fuzzy anyway. Centimorgans is another unit of length, except it's a statistical unit of length. It's not a linear unit. So instead of saying that it has 101 minus 82 mega base pairs and giving you the positional length, they're giving you an extra piece of information. They're giving you the statistical length, which is related to the statistical likelihood of a recombination event across a span of DNA. So it's actually a very useful number for predicting relationships, especially. And then the SNPs, it's the number of SNP, the number of base pairs that were actually, or bases that were actually compared to make this, to determine whether the segments match. If you got less than like 500 or something, then you want to really question whether it's a true matching segment, especially in unphased data. So for here, they all look like 7 nice segments here, and they're visualized in the form of a chart. They're represented in the form of a visual form of a chart. And you can use this information for visual phasing, no problem. People do it all the time. You can cut and paste this little chart into, say, DNA Painter, and it'll plot it on a different kind of chart for you, maybe, where you could assemble the matching segments from all kinds of different matches from different companies. This one-to-one compare tool is a very popular visual representation of segment data. On the right side is just another version of that, and I like this one because it really kind of lets you see where these are on the chromosome instead of just giving you a bunch of numbers. And I always like to visualize things. And what you have is this is just part of the data from the left, and it gives you an idea of how much of, or what percent of that chromosome is covered by the matching strand, and whether you're looking at a block or a tiny segment. For example, on chromosome 7, you might be looking at a whole block, because that looks like it's about a quarter of the chromosome, whereas on chromosome 9, you're only looking at a little segment probably. So note that the coordinates used to define the start and end locations, I'm reading off the bottom here, may vary between Jedmatch and Jedmatch Genesis, depending on your choice of coordinate system in Genesis. Jedmatch doesn't let you pick. They use build 36 coordinates and there's no way to change that. However, Jedmatch Genesis lets you decide if you want to use build 36, build 37, or build 38 coordinates. And that's just coordinates that were designed when they put all the genes in a row on a particular chromosome and tried to give each one a position based on their current understanding of how many genes are on the chromosome. Uh, and each time the 36 build 36 means it's like the 36th time they tried. It's just the version 3.6, you know. So that's what build means. And the numbers can be different, especially on chromosome 19 and some of the others. It could really screw things up when you try to do visual phasing if you're not being consistent with the coordinates. I mean, it's like if you were, you know, when you're measuring things, you want to be consistent and only use inches or only use centimeters. You know, you can't like add 2 centimeters to 2 inches and say it's 4 something or others. You want to try to be consistent. Although the difference between an inch and a centimeter is a lot different than the difference in distance in the two builds. Okay, so next one also comes from Jedmatch, and this is a more advanced tool. It's a tier 1 matching segment search tool, and it's designed specifically uh, for organizing segment data in a more useful way for visual phasing. On the left is what the diagram on the left is what actually comes out. Um, it batch processes your top matches. I think when I do it on mine, it's like my top 10,000 matches. And it figures out where all the matching segments with each of my matches are. Some of my matching cousin matches may have 10 matching segments with me. Some may have one. Some may have 40. It puts them all in order, and not by the donor, but rather it puts them in positional order. It goes by chromosome and then by start point. It sorts them all for you, and it even provides a little visualization of that on the right side, so you can see what's going on. And this is extremely, extremely, extremely useful for visual phasing because what you want is to compare other individuals and look at all the individuals that may be across a segment to determine things like, is that segment paternal? Is it maternal? Which grandparent did it come from? Do I have any known cousins that span that segment? Maybe do I have any larger matches that may be a whole block that can be used to solve an entire group of segments and things like that? Those are the types of things that this type of organization helps you with. And what I like to do is I convert it over to the right side. I take this data and I put it into Excel because then I could use some additional organizational tools that are available in Excel, like data filtering. You know, if I want to only look at chromosome 1, I could just auto filter it and look at chromosome 1. If I decide I only want to look at the really long matches, I can filter by the length and centimorgans. If I want to if I want to eliminate matches that are from a certain person that's too closely related, that's screwing up my analysis, I can filter by email and remove all of the kits that I uploaded myself, for example, or things like that. And you could also do things like color coding. And I understand that the chart on the left is color coded to a degree, but I would argue that you could do a lot more useful color coding in Excel. And you can also add extra columns for things like have you assigned phase, or any notes, or a column for does this person have a tree, or a column for does this person have a tree on ancestry.com. Maybe even though you're using Jedmatch, you may have found their tree on ancestry.com, and you might want to put a hyperlink or something. That way you can look at it later. Um, or other notes like this person has a little tiny stub of a tree, but I think I can expand it, and it would be to my benefit to do so. And you can put that in the notes column. So that's why I like to use Excel as opposed to just going directly and copying and pasting this or exporting somehow this matching segment list from Jedmatch into a program like DNA Painter. I prefer to do the intermediate step of putting in Excel first, and then of course you could also upload an Excel spreadsheet to programs like DNA Painter just as easily as you could going directly from Jedmatch. So, I'm reading the bottom again here because I pretty much explained what's at the top of this slide. Examining the overlap in matching segments between a series of donors assists in the process of identifying recombination points that divide blocks and segments. The spreadsheet on the right consists of only matches on a donor's paternal copy of the selected chromosome, so there must be a recombination point somewhere in the gap between the color-coded segments inherited from different ancestors. Why does my spreadsheet only show the matches on the donor's paternal copy? Because I'm working with phase data. And we'll get into that later. We'll get into the benefits of using phase data and I understand that's not always possible, but whenever possible, when you're visual phasing, you're going to want to use phase data, and I'll get into that. Um, and there are many ways to use phase data on Jedmatch besides using their two phasing tools, which are very nice tools. That's the phasing and the Lazarus. Well, actually three tools. They also have an evil twin tool. But there are many different third-party phasing algorithms, including, of course, my own Borland genetics ones, that can be uploaded to Jedmatch and can give you additional ways to phase relatives that are useful for the visual phasing process. We'll get into that. Okay, next visualization. I really, 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 really like DNA Painter. Um, it's kind of a newer tool. It's my favorite as far as really displaying segments and its versatility, and it's I consider it like the intermediary, the interface between my tools and Jedmatch, and I kind of consider it like a converter between other tools as well. I mean, you can show your segments from the testing companies on here, the ones all except I think Ancestry that allow you to export the segment data. But even Ancestry, you could download the file, upload it to Jedmatch, and then calculate some segment data using the matches there. Um, just make a really nice picture. And like on at the top of this diagram, it's I cut it off at the top, but it says chromosome 3 there, and it shows the paternal and maternal copies, and it matches it on the right to a legend. And then if you click on any of these segments, you can store notes in there. You can put in the exact beginning and end points. You can specify whether it's build 36 or build 37. You could have notes for the entire project. And I would say DNA Painter right now is the most versatile uh, visual representation for segment data available today, and it's free. There's a subscription service which also has some additional tools that are really really useful, like that help importing, exporting, and give some and let you do multiple profiles and things like that. And it's well worth the subscription price. But even the free copy is extremely powerful, and I think this is going to become the industry standard in terms of visualizing DNA segment data. And I'm looking forward to seeing. I know they've already had a couple versions of this. I'm looking forward to see it continue to grow over time. And okay, next slide. Next one we have is the Stephen Fox visual phasing spreadsheet, and this is really one of the pioneering tools of visual phasing. Um, it's arguably the it's the only really visualization tool that's specifically designed for visual phasing that has additional features that help you find recombination points and things like that. It's a really really nice piece of software, and I know Stephen Fox is always updating it with additional features and things like that. And this is a really nice piece of software. I have not been able to get a lot out of it personally though, because it requires um, multiple siblings, and I just don't happen to have uh, that kind of thing in my family with the DNA resources I have. But for those who do, this is really really an amazing tool, and it was one of the pioneering visual phasing tools on the internet. So I highly recommend you taking a look at this and deciding whether this is something that would be useful for you. And I also want to read the notes on the right side of this to you, because there's some important stuff on there about this type of chart. Because if you notice, it's got some elements of the Jedmatch visualization on it. It's got the blue and the green and the yellow segments, just like Jedmatch uses in the one-to-one tool in their visual representation. But it's got some other unique features here, and let's explain what they're doing. One of the most important segment visualization tools is the Stephen Fox visual phasing spreadsheet, which not only displays segment data, but also assists in identifying recombination points in the special case visual phasing workflow of comparing block data across unphased sibling DNA. This type of diagram is called a sibling parity chart. Note that the term unphased refers to the fact that both copies of each donor's chromosomes are overlaid in the analysis, so that the full siblings will have regions where they don't match at all, and we call this zero parity, where they match only on one copy of the particular chromosome in question. Here's chromosome 7, and we call that odd parity, or they match on both copies, and that's called even parity. Odd parity matching segments, more commonly referred to as half identical regions, or HIR, are identified by blue bars below the chromosome. You see a lot of blue on this map here because we're dealing with siblings, and they match a lot of places. You're going to share a lot of DNA rather. Whereas even parity segments are called FIR, and H is for half, F is for full, are shown with the long green bars, which are only a subset of those that match on at least one copy of the chromosome. Zero parity segments are the yellow with occasional red or green lines. They're the parts of the diagram that just have like grayed out on the bottom instead of a blue bar and look kind of like noise. But that's how you understand one of these, and this is a really nice tool. And I suggest if you've got some sibling data to work with, to give it a spin. It's well worth your while. But again, this is the special case that Stephen Fox spreadsheet applies to, and we're going to talk about the difference between special and general case in a bit. Other tools for visual representation of segment data. There are more out there, and there'll be plenty more to come. Even my software has a couple of tools, even though that's not really the purpose of my software. Borland genetics is kind of what you use before you visual phase and after. So it'll help you prepare the files to upload to Jedmatch, for example, for visual phasing, and then you do your visual phasing however you like, using all your favorite tools, whether it's the Stephen Fox or the DNA painter or the tier 1 matching segment or some combination of those. And then you go back to my program and you can turn those some of those nice maps that you made or nice charts that you made into actual DNA reconstruction using the raw data files. So there are many additional segment mapping and comparison tools available, including tools bundled with testing company subscriptions. Like 23andMe has chromosome browser type of stuff. So does some of the other. 23andMe even plots segment ethnicity by segment, which is kind of cool, and I really like that. And tools provided as a free or subscription products from third parties like Genome Mate Pro, which is another great tool, and I'm looking to give that a spin. I've seen a lot of what it does on Facebook, and I can't wait to try that on some of my own data, maybe over the holidays when I have a little more free time. And other products offer non-visual segment data that can be exported into DNA Painter. So, some testing companies, I think FTDNA doesn't have a particularly visual representation of their segments. They might. I don't go on there very often, and I'm not a paid subscriber to that site. But you could download segment data, in a not particularly visual form, and then you could do things like upload it to DNA Painter or things like that. So just because it doesn't have a visual representation in the program doesn't necessarily mean you can't use it to create a visual representation using some other tools. Okay. Assigning parental phase. We're getting down there to our terms in the original definition of visual phasing. Conceptually, assigning parental phase, I hope I didn't say paternal. If I did, it was a mistake. Getting late. Assigning parental phase. Conceptually, assigning parental phase is simple. For a given segment, assigning parental phase means determining whether the segment was passed down to the donor by his or her parent via that parent's father or mother. In other words, assigning parental phase is the process of identifying from which donor's grandparents a segment was inherited. Theoretically, a segment's phase is assigned by examining, I wrote phased there, it should say phase. Typically, a segment's phase is assigned by examining the cousin matches along the segment to determine what side of the family they're related to the donor. And that's visual phasing 101. A lot of people use that method, and it's a very reliable method. Sometimes this is done via family tree DNA. Via family tree data. If your match has a nice family tree available, you can do this quite easily. You can look to see how they're related to you. Just compare your tree to theirs if that's possible. But it can also be done doing shared matches by analyzing ethnicity. If you've got grandparents from different parts of the world, you may not even need donors for your trees. Just take a look, what countries they're from. Analyzing whether siblings share segment start or end locations, and that's referring to like the process in the Stephen Fox tool, and taking advantage of data peculiarities caused by the founder effect, endogamy, or by isolating segments with anomalously low match density associated with certain ethnic groups due to the unpopularity of testing in some parts of the world. So the moral of the story is that there are lots of ways and probably as many ways as you could think of. Depending on your creativity and what you know about your grandparents that might make their DNA different from another one of your grandparents, you could use that information to separate the two. If one is mostly Quaker from Ohio and the other one is a recent immigrant from Poland, you could use all kinds of things to distinguish the DNA from your four grandparents. Doesn't matter how you do it. The process of doing it is called assigning parental phase. And there we go. I think we covered all four of the elements from the definition. We'll go back to at the end to make sure we did. But um, okay, so now let's talk about visual phasing, the general case, which is what I've been talking about all along here, and the visual phasing the specific case. And when I'm making this comparison, I'm in no way saying one's better than the other. General case just means it's like general relativity and specific, and regular relativity, in physics or something. Or, it's just that one is a general case of one is a specific case of the other. Um, and the specific case is extremely useful, and the general case is more broad and it may be more complicated, but also has a wider potential application in genetic genealogy. So it's important that you know that there is a general case in addition to the very, very important specific case that is the Stephen Fox spreadsheet case. Note that nowhere, and note, I'm going to read to you now and explain if necessary. Note that nowhere in the definition of the general case of visual phasing that we started out with, right, do the terms sibling or parent or children appear. The general case of visual phasing is so labeled as general because it does not demand any specific set of DNA resources. Visual phasing is, in the general case, does not require a parent and two or more siblings. Rather, visual phasing can be accomplished using any number of combinations of DNA resources, none of which need to be siblings, in fact. So the key to a successful visual phasing project is number one, choosing the appropriate method of determining the start and end locations of your segments. To make that visualization, you need to get some solid segment data, perhaps accomplished by Jedmatch with their matching segment tools. And number two, establishing some rational criteria that can be used to assign phase. As for the second requirement, a parity chart, like the kind used in the Stephen Fox spreadsheet, can be an excellent tool for assigning phase, but it's not the only tool. It's limited in that it requires specific combinations of DNA resources like siblings. For the general case, one must apply customized criteria that suits the resources at hand, and that's really where creativity comes into play, and also just how much you know about the grandparents that you're trying to reconstruct or to map out on one's chromosomes. Using everything you know, everything at your availability, using the unique set of cousins that have tested that you have the access to their data, using unique properties of some of your cousins, like that may be double cousins or that may be their husbands may be related to the husbands of other relatives and things like that. Using what you know and what you got to the full extent is really what you need to assign phase in the general case. That doesn't mean it's harder. It's just different criteria than what we're using in the specific case. All right, now let's talk about something a little different, and that's some people mix up phasing with visual phasing, and I'm going to call, since they both have the word phasing in it, I'm going to call the ordinary sense of phasing actual phasing. So the actual phasing is, when you see the phasing tool, like for example on Jedmatch, where it allows you, if your father is tested, to phase your data and create split your data into the paternal and maternal copies using almost a process of subtraction to find to reconstruct the opposite parent. That's actual phasing. And actual phasing and visual phasing are entirely different processes. Unlike visual phasing, actual phasing involves manipulation of raw DNA data. Actual phasing is a process by which a stereo raw DNA resource, meaning it has both paternal and maternal copies, um, and those copies are overlaid. So when you open up your raw DNA file, it's not like the first column is the paternal copy and the second column is maternal. I mean, we only wish it would work that way, but you have to have phasing algorithms to actually discern which is which, and it varies on a per SNP basis. Or every base listed in there, every position, it could be one or the other, and it's completely random for our purposes. It's not completely random, but close to it. Okay, so actual phasing is a process by which a stereo raw DNA resource consisting of both paternal and maternal base data overlaid is separated into two new mono DNA resources, each consisting of either entirely paternal or entirely maternal base data. And note that I said DNA resources. I didn't say DNA kits, because it doesn't have to be an entire genome, an entire DNA kit that you phase. Phasing need not be applied to full genome resources, such as the specific case of ordinary parent phasing, where you take the entire genome of a child and you subtract out the genome of a mother and you reconstruct the father. That's you're looking at a full genome type of resource. By full genome, I mean coverage. I don't mean that they actually tested every single base pair in the genome. Phasing can be applied at the block level, and that's the type of phasing that's done in my reverse phasing process, if you've seen my video on that. Okay, it can be applied at the block level, as is done with reverse phasing, a parent using his or her child, or even at the segment level, and that's called microphasing. The latter of which, the microphasing, has applications in detecting false positive cousin matches in large data sets. So if we're talking about visual phasing, we don't care about actual phasing, right? Wrong. Actually, actual phasing is super important in the field of visual phasing, or when you're trying to accomplish visual phasing. And here is why. Actual phasing, and this is before you do your visual phasing analysis, can turn a 2-week visual phasing workflow into a 6-hour workflow. It's that much different, and it makes it that much easier. Okay, let's talk about why. I think I got like 7 reasons here. A phased genome has half as many recombination points to locate and analyze, because visual phasing is applied to 23 chromosomes rather than 46, and that's just because you have, it only looks at one copy at a time, and you may end up doing the other 23, but you've got a finished product when you're done with 23. When you're done with the first 23, that could be used for reconstructions or whatever you're going to do with your visual phasing map when you're done with it. You're going to be done in half the time already, because you only have half as much work to do. You only have to find the recombinations on 23 chromosomes, not 46. Number 2, assigning parental phase is simplified because there's only 2 grandparents to choose from rather than 4. I mean, if you're looking at only one copy of the chromosome, you're eliminating data from 2 of those grandparents. So you don't have to decide whether it's grandpa, grandma 1, grandpa or grandma 2. You just got one set of grandparents, and there's less choices, so there's less confusion. Like, let's say you had let's say both your paternal grandfather was Polish and so was your maternal grandmother. You don't got to figure out whether the Polish segment that you're looking at belongs to one on the father's side versus one on the mother's side, because you're only looking at 2 grandparents at the time. And if your paternal grandparents, one is Polish and the other one's Lithuanian, then when you see the Polish one, you're probably looking at which grandparent you're looking at. Maybe that's a bad example because they're neighbors on the map, but I think you see what I'm saying. Number 3, pinpointing recombination point locations is much easier, as endpoints aren't as fuzzy when dealing with phase data. And we've talked about that. And the reason is because they're not overlaid on top of each other in the file, therefore creating a mess. For most SNPs, you only have 2 possible choices of bases. And if you got 2 different people, I mean, a lot of the times, even if you have perfectly good measurements of those, it may as well be a no call for the purpose of determining whether it's part of a segment matches. Because if you got both grounds covered, they're going to match everybody, right? I mean, if you got a C from your father and a T from your mother at a particular location, and everyone in the world either has a C or a T on every chromosome, I mean, you're going to match with everyone at that point. So that's why you get a lot more fuzzy long ends after the tips of true matching segments when you're dealing with data that is not phased. So phase the data. Number 4, there are far less false positive matches that have the potential to throw the process off track, as phasing greatly reduces the threshold at which a cousin match can be trusted as inherited by descent. And that's for the same phenomenon as the fuzziness of unphased matching. It's because of the overlay. That overlay creates tons of false matches, especially when you get down to like the 7 to 10 centimorgan range in terms of the size of a segment. The phasing just corrects. When you look at compare phase data, those matches will disappear. Those cousins, if they're not real cousins. Generally, when I'm working with phase data, doing visual phasing, sometimes if it's depending on what my resources are, I may lower the threshold down to 5 centimorgans even. We're not lowering it down to 1 or anything like that. That's absolutely ridiculous. You won't get any real information out of that. But say where like the absolute minimum you'd ever want to work with unphased data is 7, with phased data, it might be 5. Okay, the sibling parity charts, if applicable, and that's here we're talking about the Stephen Fox stuff, only have 2 modes rather than 3. Because I think you can use phased data in the Stephen Fox spreadsheet, although I haven't actually tried it. Like I said, because I just don't have many siblings. So what happens if you're only using phased siblings in those? You only get 2 possibilities. You got 0 parity and you got odd parity, because there would be parts where they match on both copies, because we're only comparing people. We're only comparing 1 copy of the chromosome at a time. So that's one less scenario you have to bother with trying to figure out what the heck to do with when you're using the Stephen Fox spreadsheet. So phasing has also practically eliminates the instances where recombination points are overlooked for being too close in proximity to one another. And that's an issue that I see a lot of people in the visual phasing work group on Facebook having an issue with, determining, well, gee, was this a recombination point? It was so close to the other one. But you're working with unphased data. If you were working with phased data, you'd have half as many recombination points. And like magic, the ones that are really close together that you're not really sure if they're recombination points, they're not going to be there, because one of those recombinations was probably on the paternal copy and one on the maternal, and we phased, and we're only looking at one at a time. So that really eliminates, or it doesn't eliminate, but it reduces the amount of hassle that you have to spend dealing on figuring out whether potential recombination points that are close together are true recombination points, or just a blurb of green on your thing by accident because maybe your parents are distantly related. They're 20th cousins. Okay. When phase must be assigned by stranger matches, and those are cousins of unknown relation, and maybe they do or don't have trees, one need not triangulate to determine whether the stranger is paternal or maternal. Because I see a lot of time people are using unphased data, spend a lot of time triangulating, when that's pretty much already done when you phase your data. There's no need to triangulate to figure out which copy of the chromosome you're working on, because you're only working on 1 copy at a time. So all the stranger matches to a phase kit are either paternal if it's a paternally phased kit, or maternal if it's maternally phased kit. And there's no worrying about triangulation. Okay, the layers of and this is an important one. The layers of complexity removed by visual phasing using actually phased kits allows for the application of visual phasing in the general case to a wider variety of DNA resource combinations with ease. So it really takes away the limitation of only using the sibling special case, the Stephen Fox case, and really opens the door to a wider possibility of just what relatives you can use for a visual phasing project. I mean, the last one I did had 13 DNA kits from a family reunion, and among those 13, only 2 of them were siblings to one another, and there was a pair of half siblings also. All the rest were just randomly related cousins, and I was still able to do a successful reconstruction of our common ancestors. And one final advantage for using phase data and visual phasing projects is you can use if you're using phase data, that you can then use my Borland Genetics free toolkit easily to dissect the phase data. I've created all kinds of algorithms to reconstruct synthetic DNA kits for your grandparents, for your ancestors. If you did the visual phasing with phase data, then I've got all kinds of way to realign the blocks and segments in meaningful ways to reconstruct your ancestors. And my tools are free, so I just want to get this out here because I think we can do a lot of good visual phasing work. And there'll be a lot more synthetic reconstructed ancestor kits for people that the community can derive a lot of value from. If we just broaden our horizon a little bit about what visual phasing is. And I'm going to I said I'm going to make a series of videos because there's an equal, I think, issue with phasing itself, with actual phasing. I want to make a video on the special case of parent phasing and how that they're the same type of thing, a general case phasing video to explain that that's really not the only kind of phasing there is. You can do all kinds of phasing. You can phase just about any two types of relatives for useful purposes. Um, and the third video, I guess, maybe a combination of those and how that you use these together with my tools and how you can really get a lot out of taking making workflows that involve general case visual phasing and general case phasing to reconstruct your ancestors from 1700s, 1800s, actually creating DNA kits for them, and not just a Lazarus kit with like two segments from an ancestor that was born in the 1700s, but rather a decent coverage, decent resolution. It won't be 100, of course, it won't be 100%, but um, nice reconstructions that can go on Jedmatch and generate cousin lists just like as if they had tested today.